That was such a powerful introduction. I just wanted to thank Ralph. And I guess start off where he left off a little bit. So I think one of the points that he emphasized was that people tend to think that lead is a solved problem. I mean, we did have a lot of regulatory effort back starting in the 70s, 80s, and 90s. We eliminated lead from gasoline in 1996, so that's 20 years ago. We eliminated lead from new household paint in 1978. And there has been some success in reducing lead levels. So nationally, the percent of preschool-aged children with lead levels over 10 micrograms per deciliter fell from 8.6% in 1998, which is pretty horrifying, to 1.3% in 2004. But the flip side of this is that a lot of people seem to think that lead is no longer a problem, whereas the CDC says that 4.5 million households in the US are still exposed to high levels of lead. And also disadvantaged children and African American children are much more likely to be exposed. So for African American children, the rates of lead poisoning are 50% higher than for other children. For low-income children, such as children who qualify for National School Lunch, the rates are 30% higher for low-income children. And when we're talking about lead now, we're typically talking about much lower levels than 10 micrograms per deciliter. So what I'm going to talk about today is some joint work with Anna Azer, Peter Simon, who's here somewhere, yes, and Patrick Vivier from Brown, looking at lead poisoning in Rhode Island and how that's related to school performance about kids in Rhode Island. And one of the great things about Rhode Island is that they have a very robust program of testing for lead. So Ralph was talking about how in New York, there's still a lot of kids that don't get tested. In Rhode Island, I guess because they have a lot of old housing and there's been a lot of focus on this problem, 80% of kids get tested by the time they get to school. So we were able, with a lot of assistance from Patrick and Peter, to get various agencies of Rhode Island to work together to link administrative data. These administrative data that we're using for these studies are data that every state has. So this kind of study could be done in any state where there was cooperation between agencies, which is not a small ask. But so the Department of Health has data on child lead levels from their lead testing program. And so for the children, they have the date of the test, the location of the child, and the method of the test, whether it's capillary or venous, and also the blood lead level. And in Rhode Island, the kids get tested multiple times, so the median child is tested 4 times, which is very helpful in coming up with an accurate measure of their exposure. We linked that to data from the Rhode Island Department of Education, which has their test scores for reading and math in grades 3 through grade 8. And today I'm mostly going to be talking about 3rd grade test scores. Also, we know whether they were eligible for free lunch, whether they had an individualized education program, and whether they have a disciplinary infraction of some sort on their record. And then we further linked that to Rhode Island vital statistics data, that is birth certificates. And the reason for doing that particular linkage is the birth certificates have a lot of information about the family. So you know things like the mother's education, and also when they got prenatal care, whether the child was low birth weight, and so on. So by linking all that data together, we have a very powerful way to look into this issue. And then the fourth thing, I guess, about Rhode Island that was really useful was that they had an aggressive program of lead remediation. So I'll talk about that a little bit. In 1997, they announced new policies to reduce lead poisoning. This was in response to a call from the CDC that states should really do something about this. And so what they came up with were lead-safe certificates for landlords. So landlords were required to obtain a lead-safe certificate in order to rent housing. So again, it was focused on rental housing mainly. It targeted particularly areas that were known to have a big lead problem. And it offered not so much sticks as carrots to landlords. So landlords were given information about how to do lead remediation in a low-cost way. They were given interest-free loans, if necessary, in order to carry out the remediation. And by 2004, the end of our sample period, the number of certificates had gone from essentially 0 to 41,000. Okay, so there were a lot of people taking part in this program. So what I want to show you is what the effect of that was. Okay, so here's a figure showing the average preschool lead level by birth cohort. So remember, this is essentially all the kids born in Rhode Island in each year between 1998 and 2004. And in this 6-year period, which is a fairly short period to accomplish a change, the blood lead level, on average, went down from about 4 micrograms per deciliter to something in the order of 2.5 micrograms per deciliter. Okay? So we're talking about pretty low levels, and we're talking about a big percentage change, although it's essentially 1.5 milligrams per deciliter reduction in blood lead levels. Okay. Looking at that in a little bit more detail, you can look at the whole distribution of blood lead levels. So what you see in this figure along the bottom axis is the average lead level, and the blue line shows what the distribution looked like in 1998. So you can see if you focus on that group that gets more of the attention, people with 5 to 10 micrograms per deciliter, or even higher than that, there were a significant number of children in that category in 1998. Okay. By 2004, which is the red line, the whole distribution has shifted downwards, thankfully. But you can see there's still sort of a mass of kids with 2 or 3 micrograms per deciliter in their blood by 2004. Okay, so there's an improvement, and a lot of it is at this low end of the lead exposure distribution. If you look by characteristics of the children, again, you get a somewhat more nuanced picture. So here, African Americans are with blue dots in the first picture. Caucasians are red dots. And so you can see that for both groups, lead levels are going down, but they're going down much more for African Americans. That's partly because African Americans had higher levels to begin with. So if you're going to improve things, it's actually going to probably have a disproportionate impact on African Americans. The second graph that's labeled by income is looking at actually free lunch versus not free lunch, because that's what we have as a measure of income. And you can see there, again, that the kids who qualify for free lunch have much higher blood lead levels, both at the beginning and the end of the period, though the blood lead levels are kind of converging for the two groups. Okay, so what does this have to do with test scores? This figure is just showing, on average, what is the relationship between these blood lead levels, test scores, and also I threw in here at the bottom right, maternal education. Okay? And I threw that in for a reason. So, but you can see these lines are downward sloping for the two test scores, for reading and math scores. The higher your lead level, the lower your score. Okay? And even for kids with less than 5 micrograms per deciliter, that's still true. There's a pretty steep decline there for each unit of lead in your test score. Okay? The first graph on the bottom is looking at whether you have an IEP. That's going up with lead. Okay? Now, the last graph for maternal education is showing, oh, but mother's education is going down. This is mother's education at the time of the birth, is going down with child's lead level. And that sort of illustrates what the problem is with making inference here, right? Because the child's blood lead level couldn't possibly have affected the mother's education. So that can't be a causal relationship, but it's showing that less educated mothers are more likely to have kids that have higher blood lead levels. Okay, so that's where the program comes in. By using the fact that there was a big lead cleanup, we can say, okay, here's a outside change in lead levels, and let's see what's the effect of that, because that didn't really have anything to do with anybody's socioeconomic status. Okay, so when we do that, what we see is a big effect on the probability that the child's reading proficiency was deemed to be substantially below proficient. Okay? So about 21% of the sample kids had reading scores that were below proficiency. A unit increase in lead is associated with about a third increase in the probability of being below proficient in reading. Okay. For math, you see the same thing, basically. For just a 1 unit increase, a increase in the probability of being below proficient by a third. Okay, so these are really big effects on children's outcomes from what seem to be fairly small changes in lead. So this really is a very toxic substance. Okay. Looking, just to summarize again, what happened to the reading scores for these 2 cohorts. You can, I've done it by race because I think it's very striking. You can see that for white kids, the reading scores increased a little bit. The whole distribution shifts a little bit if you compare the blue and the red lines. But for the African American kids, there's really a big improvement. Okay? And so you might say, well, you know, maybe there was some other program in Rhode Island over this exact period that caused there to be a big improvement in reading scores for African American kids. And that's possible. But we get actually the same results as these if we look at kids living in the same house before and after the lead cleanup. If we put in controls for the test scores of all the other children in the same school and class. And also if we allow there to be different time trends going on for African American and other children. And so we think that this is actually a causal effect that we're measuring here. Okay, so I want to talk a little bit about cost effectiveness, because when you're talking about policy, that's always an issue. And I want to point out that this was an extremely cost-effective program. The cost per child worked out to something like $679. We judged that it improved test scores by about 1/4 of a standard deviation. So those of you in the education world will know that's a fairly decent-sized effect. And relative to other educational interventions that we know about, things like radically reducing class size, this was extremely cost-effective. Okay. So just to summarize, we are arguing in this study that lead has a negative and causal impact on cognitive functioning, as measured by test scores, even at these extremely, by historical standards, low levels. This particular lead policy, which was aimed at reducing exposure, disproportionately lowered blood lead levels of low-income and African American children and reduced gaps in test scores. The reductions were associated with improvements in reading and math test scores on the order of 1/3 reduction in the number of children being judged below proficient in math and reading. I haven't shown you these results, but it was also associated with reductions in disciplinary infractions by about 5%. So overall, it was an inexpensive, extremely effective policy which had real impacts on children in schools. I'll quit there.
I want to, first of all, thank the organizers for inviting me. Excuse me? Oh, sorry. I want to thank the organizers for inviting me to participate in this very interesting and important meeting today. And I'm going to talk to you about some of the what we've been learning about the biological effects, particularly of low-level exposures, and what lead is actually doing to the brain, how extraordinarily harmful it is on a whole bunch of different levels, and how we think that is contributing to abnormal brain functioning and what's happening to these kids in the school system. So first I'm just going to talk a little bit about lead and where we're finding it and some of the effects that it has on children's health. So we all know that deteriorating paint that contains lead is a huge potential hazard for these kids. But lead crops up in sometimes unexpected places. It's in some candies and spices that are brought in from other countries. It's in paint and materials made to produce certain toys that are also imported into this country. It can be in water supply, both in the home and in the school and in other public places. It can be in the soil where kids are playing. So there's lots of different routes of exposure. And lead not only can affect brain structure and function, but it can influence hormone levels. It can cause significant cardiovascular problems later in life. It can produce anemia. It can harm the kidneys, cause a variety of gastrointestinal problems, affect the functioning of the reproductive system, and even influence growth due to its long-term storage in bones. So its effects are harmful on a variety of different levels and can cause a lot of problems, not only during childhood, but later in life. So developmental exposure to lead, we now know has extensive effects on the nervous system. It, even at very low levels of exposure, it adversely affects both the structure and the function of the brain. And it influences activities at all different parts of the nervous system. In neurons, the cells that actually transmit information through the nervous system, it can influence not only the structure of these cells, but the production of neurochemicals that are critical for transmitting information through the brain. It can affect the growth and health of cells called oligodendrocytes that produce the myelin sheath, or the insulating material that goes around axons or nerve fibers, and if there's any breaks or problems with that myelin or insulating material, it's kind of like breaking the insulation in an electrical wire, and the information just is not transmitted effectively through the nervous system. It influences transmission of information at what are called synapses. Those are the spaces where signals transfer from one nerve cell to another, and the way that information is transferred is seriously disrupted by lead. And also these other cells called astrocytes, which are support cells for the nervous system. These cells are important for bringing nutrients from the blood system to the nerve cells and for eliminating metabolic waste products from the nervous system, and the health and functioning of these cells are also disrupted. So lead can influence all the different cell types and innumerable functions of all of these cells. At a very basic level, it interferes with the transcription of genes, the production and expression of a variety of different proteins and a large number of both molecular and cellular processes that adversely affect the growth and development of the nervous system. So as a brain is maturing, and these are some pictures from the cells in the cerebral cortex of a newborn, a 3-month-old, and a 2-year-old. And what you see is that the growth of neurons, there's more neurons that are recruited into the cortex that migrate in there over time. And not only are there more neurons, but the complexity of the interactions between these neurons increases dramatically as the brain is maturing. And this increase in complexity of these neural connections is critical to the normal development and maturation of the brain, and this is what leads to the normal development of cognitive functions. And what we found a number of years ago when we wanted to look directly on and whether or not lead has an effect on this sort of complexity of these interactions between nerve cells, we looked in a very straightforward way by growing nerve cells in a dish in the laboratory and then exposing them to various levels of lead. And what we found was as we increased the amount of lead that was added to the environment of these cells, we didn't actually kill off nerve cells. We didn't have fewer cells, but what happened was there was a dramatic decrease in the length of these processes on the cells and a tremendous decrease in the complexity of these interactive circuits between these cells. And as you see down here at the bottom, there's very stunted little processes coming off these cells, and that's with a lead level that would be equivalent to 0.24 micrograms per deciliter. Now we're currently using 5 micrograms per deciliter as our reference point for lead poisoning. So at a fraction of that amount of lead, you can see that there are just tremendous effects on the development of the structural integrity of these neurons and their circuits. So the result of this in a developing brain is that it has a tremendous potential to be improperly wired as the brain is being put together. So what is the potential real-life implication of this type of study where we're looking at isolated cells and culture? Well, there was an interesting study that was published by Kim Cecil's group several years ago where they showed that there was decreased brain volume in a number of different brain regions with an inverse dose-effect relationship between childhood blood lead concentration and brain volume, such that the higher the childhood blood lead levels were, the more there was loss of brain volume. Now, decreased brain volume can be due to a variety of different factors, and one is that you just lose neurons, and this happens in certain neurodegenerative disorders, for example, in Alzheimer's disease, where you're losing cells and you get brain atrophy. But what we see in these brains of people, these are adults who were exposed as children, is not that we see atrophy, a lot of very stark atrophy, which would suggest the loss of neurons. We still see this loss of brain volume, which more likely, I think, reflects a loss of this complex network of axonal, dendritic, and glial branchings that form the bulk of the gray matter, which is what I was showing you on that previous slide, that series of interconnections, all those fibers. So I think that's what we're losing in the human brain, which would be precisely what we were seeing when we exposed these cells in culture to lead. Interestingly, in this clinical study, the effects were greater in males and females, and I'm going to talk a little bit shortly I'm going to talk a little bit more about what we find experimentally as a very significant sexual dimorphism, that lead affects female brains differently than it affects male brains. So what are the functional implications of these kinds of disruptions to the structural integrity of the brain? Well, this was a functional magnetic resonance imaging study that was published by Wan's group in 2006. And what it shows is that childhood lead exposure had a significant and persistent effect on the functional organization of the brain. Now, this was a study that was done in adults, again, who were lead exposed as children. And what you see here is that in this particular study, where the subjects were asked to perform a verbal word generation task while they were in the scanner, the subjects who had higher lifetime mean blood lead exposures had less activation of the brain than subjects who had lower lifetime exposures. Now, we've done some preliminary studies in adolescents who were exposed to lead when they were younger, and we've found something very different. And it's possibly because we're looking at a different type of cognitive function, and one that is actually quite difficult and taxing. So in our task, we had the adolescents in the scanner, and they had to perform something called a two-back test. So this is a fairly difficult test of non-spatial working memory. So what they had to do was, while they're lying in the scanner, they're looking at a screen, and they have a little button and switch attached to their hand, and they have to watch the screen for letters of the alphabet to flash on the screen. And when they see a letter that's the same as a letter they saw 2 letters ago, they have to move their finger and touch the switch. So you have to keep updating your memory, and you have to be paying attention. So this is a pretty difficult task, I know, because I tried doing it and wasn't all that great at it, which I attribute to age, but hopefully not to any other brain damage. But what we found was really very interesting, in that the group that had the higher level of lead exposure had greater signal in their brain, had more signal in their brain than the subjects who had the lower exposure, which was fascinating, and to me means that those brains are needing to recruit more brain circuits and needing to pull in more of the brain to try to do that task than the less damaged brain. That there's a greater resource utilization in individuals with higher prior blood lead levels. So what I think this means is that this reflects a need for the injured brain, the injured memory circuits, to recruit additional neural resources in order to meet the demands of this very challenging task. And therefore, a decrease in, or the decreased activation, we think probably reflects more efficient brain processing. This increased activation pattern reflects less efficient brain processing of information. So individuals with lead exposure then may need to use different cognitive processing strategies to perform a working memory task like this, and probably other tasks as well, than individuals with lower lead exposures. And this could have, you can imagine how this could have overwhelming consequences in classroom performance. So one of the other things that we've been doing is looking at lead effects at the gene level in the brain. What is lead doing, and how extensive are its effects on expression of genes that could be involved in a variety of different functions, including learning, memory, and other cognitive functions. So to do this, we expose animals to lead, and then we take out parts of the brain, and we extract the RNA from these parts of the brain, and we run them on something called a gene expression microarray, which is shown here. It's what you get is a bunch of little dots that some are green, which mean that if a dot on the array turns green, it means the gene was downregulated. If it turns red, it means the expression of the gene was upregulated, and if it stays black, it means it hasn't changed. And we can look at tens of thousands of genes at the same time. And one of the first things we found was that developmental exposure to lead was altering gene expression patterns in critical brain structures in a sex-dependent manner. And we found that there are some genes that are increased in expression by lead in both males and females. Some are decreased both in males and females. But we found, interestingly, in some sets of genes that are increased in the male brain and decreased in the female brain, and some that are decreased increased and decreased in the opposite directions. So that was actually the first time that was reported, and that led us to look at some other differences in expression, and we found that not only are these gene expression patterns modified by sex, but also by the exposure level, how much lead the animals were exposed to, and when during development they were exposed. So you get very different effects on the brain, not only if you're male or female, but also when during development you were exposed. Some animals were exposed in utero, some in the early postnatal period, some in a later postnatal period. And depending upon when during development you were exposed, you're going to see very different effects on gene expression. And those differences are also expressed behaviorally. So when we look at animals doing what we call an associative learning and memory task, we find also sex-related differences in their behavior, just like we saw sex-related differences in their gene expression patterns. So interestingly, for example, in this particular behavioral paradigm, where we're looking at memory of an association that they were taught previously 1 day, 2 day, and this is 10 days after the learning, we see that in females that were exposed to lead during an early postnatal period, their memory is impaired. Their recall of the information they learned is deficient. If we look at males, we find that the males that had early postnatal exposure are doing fine, but males that were exposed perinatally during gestation and the early postnatal period are the animals that had differences. So again, not only do the differences that we see, so basically what we're seeing in our gene expression patterns does translate into behavior in terms of the differences that can occur in the brain, not just due to sex, but also to when during development your brain was exposed to lead, because different things are happening developmentally in your brain at different time periods. And so, not surprisingly, different processes of brain development will be affected depending upon when and for how long that lead was present. So another thing that we're looking at is something called epigenetics, which epigenetics refers to a process through which your DNA can be chemically modified to either make genes more active or less active. So epigenetic changes can influence the way genes are going to be expressed. And epigenetic mechanisms can be affected by a host of different factors and processes, including environmental chemicals, different drugs, pharmaceuticals, stress, diet. There's a lot of factors that can affect epigenetic processes, which are basically two main categories of chemical changes that occur to your DNA. One is called DNA methylation, where something called a methyl group can attach to your DNA, and depending upon the methylation state of your DNA, it can either cause the DNA to be all kind of wound up and genes to not be accessible, or it can relax the DNA and genes can be accessible. So it can either promote gene expression or inhibit gene expression. And another chemical modification is called acetylation, where another type of chemical called an acetyl group can do similar kinds of things to the DNA. Well, what we're beginning to find is that lead exposure, developmental lead exposure, can influence the methylation patterns of the genome and can also affect the acetylation states of the genome. In some of the early studies that we've done, we've found that looking at across the entire genome, that there are literally thousands of differentially methylated regions across different developmental periods that we've looked at due to exposure to lead, and also is modified by sex. And the frightening thing was that overall, at lower levels of lead exposure, we saw greater numbers of differentially methylated regions across the genome. What's now even more frightening is that there's some preliminary data through studies that we're doing with some collaborators where we're looking at potential transgenerational inheritance of aberrant epigenetic marks. So there is the potential for changes in the epigenome to be transmitted transgenerationally. And we're now beginning to look to see if such changes in the epigenome are occurring as a result of developmental lead exposure. So although the methylation data we have shows that there's some aberrant DNA methylation profiles in lead-exposed animals, it's really not clear at this point as to how these changes in DNA methylation contribute to specific learning and memory problems, but that's something that we need to look at. Also, it's important to note that there's a dynamic regulation of these epigenetic signatures that occur in the brain with learning and memory, and our preliminary data suggests that exposure to lead blunts the ability of the brain to mount these dynamic regulation events so that DNA is abnormally methylated or demethylated during learning and memory in a brain that has been exposed to lead. So there's a variety of factors that can significantly modify the influence of environmental exposures on a brain. And some of them are listed on this slide. There can be a variety of developmental risks, stress, smoking, socioeconomic status, physical and intellectual activity, your sex, diet, nutrition. But one of the things that is sort of the wild card in all this is your genetic background, which really means that most, if not all, of these risk factors are going to be unique to each individual. And we've seen that even in our experimental studies where we've looked at whether genetic background can modify the effects of lead on gene expression patterns and behavior in the brain, and in fact, we found that they do. By looking at animals with very different genetic backgrounds, 3 different genetic strains of animals, we found that the gene expression patterns were widely different, that one strain is very sensitive, has over 900 genes affected by the lead exposure. Another strain was moderately sensitive and had about 300 or so genes affected, and another strain was relatively insensitive and had only a few genes affected. And these are animals that were exposed to lead in the 2 to 5 microgram per deciliter range. So clearly, your own particular genetic background is going to have a great influence on modifying the effects of lead on the brain. So what does all this mean for a lead-poisoned child and his or her ability to learn and achieve in school? As Ralph mentioned in his talk, lead poisoning really results in an acquired brain injury. And each child is different, but there is typically a cluster of cognitive behavioral problems that exist. And these problems are going to be determined and expressed as a factor of how lead has affected that particular brain and which parts of the brain. The other thing we know is that lead is not going to affect the whole brain in the same way. So certain parts of the brain are going to be affected more than others, and we find that frontal lobes in particular are quite vulnerable to lead and produce a variety of, or interfere with, when the frontal lobe functioning is impaired by lead, it results in a variety of clinical features like lack of motivation, inability to plan, poor judgment, aggression, difficulty staying on task, all the kinds of things that contribute to poor performance in the classroom. So what are some of the issues regarding effectively educating the lead-poisoned child? Well, we need to better understand what their deficits are and improve the way that we teach them. And importantly, I think we need to develop evidence-based recommendations for helping lead-poisoned children in the academic environment. We can't guess at what we think is going to work for them. We've got to do the research. We need to do rigorous, scientifically valid clinical research to understand what strategies may or may not work. And we have to understand that effective management is going to require an individualized approach for each lead-poisoned student because each child is going to be different. They're going to have different problems. They're going to have different strengths and weaknesses, and that has to be taken into account. So to improve educational outcomes for lead-poisoned children, we need to identify the cognitive problems in each child. And you need to do that through a well-constructed neuropsychological evaluation. And Dr. Litsky, I know, is going to talk about this at length. We need to design programs to enhance a lead-poisoned child's capacity to process information more effectively, because we know their brains are not processing that information the way it should, and improve their adaptive functioning. And the last thing is we need to try to see if we can stimulate plasticity, and to what extent stimulating plasticity is going to be possible in a lead-poisoned brain. Now, we've seen in our gene expression studies that a number of genes that are critical for brain plasticity are impaired as a consequence of developmental lead poisoning. So we need to know if this is translatable to the human. To what extent may the potential for plasticity be blunted in a lead-exposed brain? And we don't know that yet, but we need to do the research. We can't just guess at it. Because if we can stimulate plasticity in the brain, there are ways potentially that we can improve the cognitive outcomes and the educational outcomes, and that is by utilizing some of the emerging concepts in cognitive training and cognitive plasticity that have been used in other populations with other types of brain injury to improve outcomes. So remember I showed you that earlier on, I showed you that functional MRI data where we saw that increase in, that abnormal increase in signal in children who are trying to do that working memory task. Well, other people have found similar things in other types of brain injury where, again, there's this increased resource utilization of brain in the damaged brain. And if you train that brain with specific cognitive exercises, you can see that you can reduce that abnormal resource utilization and bring it to a more normal pattern. So the question is, to what extent can we do that in a lead-poisoned brain? And if we can do that, if that brain is amenable to these cognitive training paradigms, and we can show that we can change the abnormal connectivity patterns in the lead-exposed brain, we might be able to improve the cognitive outcomes of these kids and improve their school performance and give them a better chance at success in life. So, yeah, these kids are failing, but we've also failed these kids, and we've got to do a better job because we obviously have to try to prevent lead poisoning, but there's so many kids out there that have already been poisoned, and we've got to do something for them. And we've got to change the way we've been going about this because what we've been doing just isn't working. We've got to do better, but we need the resources to do the research so that we can help them. Thank you. On a regular slideshow. Just wait. So that way you can see what's coming. Yeah. All right.
All right, this is kind of a continuation of Jay's talk. The fact that one of the primary difficulties that schools have in dealing with lead-poisoned children is the fact that they're brain damaged. And the question is, how do you assess a brain-damaged child to come up with a plan to help that child? Which is this is advanced? Oh, no, okay, great. This is kind of a very simplified flow diagram of various solutions. Primary prevention, which means eliminating lead, has been advocated since at least 1991 by the CDC. That would solve the problem. However, it's enormously complicated, it's enormously expensive, and though we've made advances, there's a long way to go. Jay talked about the necessity to try to do research on whether there's a possibility of plasticity in a lead-poisoned brain. I did a search of the NIH database, and of thousands upon thousands of contracts and grants that NIH funds, there were exactly 6 grants on childhood lead poisoning. None of them had to do with treatment, not a one. Okay, so we're left basically with Ralph Spezio's problem, is picking up the pieces after the child's been damaged. So basically, we have to intervene, and the intervention depends upon understanding the cognitive effects of lead. This is something that people I'm sure are very familiar with. Lead affects IQ. Normal IQ for the population, the average IQ is 100, which is 50th percentile. With an increase of blood lead to 10 micrograms per deciliter, you lose on average about 7 points. Another 10 micrograms per deciliter, you lose about another 3, and so on, going on up. Some children it's less, some children it's more. There are various risk factors. But there's a problem in that IQ loss, as Ralph pointed out, is really the tip of the iceberg. IQ really doesn't tell you very much, and it's not very sensitive to brain injury. Although it's counterintuitive, you realize it's based on that. If you look at this diagram, Jay showed the same one. This shows it's a group MRI showing the effects of lead on a cohort of young adults with poisonous children. The areas where it's orange or red is where there's either reduced tissue volume, which could be due to loss of tissue, or loss of complexity, or so on. But it affects different parts of the brain. Other parts of the brain are left more or less intact. Different parts of the brain do different things. When there's a brain injury, usually you see a deficit in that function that's mediated by the damaged area. Functions that are not mediated by those areas are more or less intact. IQ is a single score based upon multiple subtests that tap in multiple individual unrelated functions. So if you have a weakness in one function due to a focal brain injury, it gets washed out by normal functioning in the others, and while you may lose some IQ points, it doesn't really show the significance of the problem. This is another problem with IQ tests is they don't test important parts, functions of language, certain parts of memory, attention, executive functioning are all not tapped or poorly measured by IQ. This is illustrated pretty well. This is from my experience. I evaluated about somewhere between 1,000 and 1,500 lead-poisoned children over the years. This is a child with an IQ of 100, right at the mean for the population. That corresponds to the 50th percentile. It's a dotted line. And these bars correspond to different functions. For example, this is working memory, which is normal, what you'd expect for a child with an IQ of 100. These are various aspects of executive functioning, all more or less normal. This is language. Both receptive and expressive language are very, very low. This is an indication of brain injury. Both verbal memory and visual memory are also extremely low. So while this child's IQ is 100, and he couldn't get special education because of that fact he had so-called normal IQ, he had virtually no memory and very, very poor expressive and receptive language. IQ clearly does not show the significance of the problem. So what do? How do you measure? The way you measure is something called neuropsychological testing. Unlike IQ testing, neuropsychological tests are very, very tightly focused. They target individual functions that are mediated by specific brain areas. They're objective. They're not like Rorschach tests or ink blot tests. They're valid in the sense that they measure what they're supposed to measure. A test of memory doesn't really rely on fine motor functioning or other functions that complicate the results. They're reliable, which means that if you give the test or I give the test, as long as we follow the procedures in the manual and score it the way the manual says to score it, it should have the same score. And there's a reference baseline, so you can compare that person's performance to other people to see where they stack up. Neuropsychological tests don't assess information learned in school. There are no reading tests, no tests of math or arithmetic, nothing in science, history, or social studies, or anything else. It assesses not what was learned, but what the tools that the person has to learn with. Neuropsychological functions are broken up into very broad domains: fine motor functioning, attention, memory and learning, executive functions. Executive functions are really a sort of an umbrella term for what is considered to be higher functions such as abstract reasoning, concept formation, planning, the ability to adapt to change. Memory and learning, there's verbal memory, there's visual memory, working memory, short-term memory, long-term memory. There are individual tests for all of these different functions. With attention, there are various aspects of attention, all mediated by different brain systems. There's the ability to pay attention to what you see versus the ability to pay attention to what you hear. There's sort of selective auditory attention, so if it's called the cocktail party effect. If you're talking to somebody who's very boring, and someone over there is talking something that's very interesting, you shut this person out and focus over there. That's another function of attention. The individual tests for all of this. I'm going to show you some examples of some neuropsychological tests and a couple of instances how lead-poisoned children do on those tests. Right, this is normal visual spatial construction. If you look in the middle of that figure, there's a figure called the Ray-Osterrice complex figure. Child's task is simply to draw it. Right? What you see on the left on top is that normal performance of a 6-year-old. To the right is an 8-year-old, pretty impressive by a 10, and by the time a child is about 12, it's very, very accurate. This test is not simply a test of drawing ability, it's really a test of organizational and perceptual skills. This figure is also used for assessing visual memory. Child is given this figure to draw, and then after the child draws the figure and the drawing are taken away. You talk to them for about 3 minutes, and then you ask the child to draw it from memory. And here's what you get. On the left is a complex figure. In the middle is the normal performance of a 12-year-old. This is 3 minutes after she drew that figure. And to the right is a typical performance of a lead-poisoned child, same age. This is an example of a test of verbal memory. Take a minute just to read this paragraph. This paragraph is read to the child at a relatively slow pace. And then immediately afterwards, the child's asked to repeat back whatever they can remember. Now, this is the child I showed earlier with the IQ of 100. What he said after stories read to him was, a fourth grader went to Disneyland. That's all he remembered. That's with an average IQ, an IQ of 100. All neurocognitive functions are at risk with lead poisoning. Attention, memory, executive functioning. There is no signature injury. Something that Jay pointed out with another name simply means that each child is different. Signature injury is when if a child presents to you without any history, you know nothing about the child other than the neuropsychological test results, you can say based on the pattern of results, you can identify the cause of the problem. That's called a signature injury. Lead does not have a signature injury. I'll show you an example of that in a minute. There's something also called the lag effect with lead, in that lead's effects develop slowly over time. A child can be normal, if a child, say, poisoned at 2.5 years old, can be normal at 5 and show problems at 7 or 8, or show problems at 10 or 12. Right? A child who is not normal at 5 can get progressively worse. You just don't know. But the lag effect affects about a third of the children who are lead poisoned, in that the new impairments develop over time, or existing impairments get worse. And finally, social judgment and emotional development are also at risk. They're also cognitive functions. I'll give you examples of all of this. All right, each line here is one child. Right, this is going horizontally across. First one is a child with a peak blood level of 5. And this child had problems in visual memory only. If you go down the list, these are just the peak blood levels, so the maximum was 15. You see, each child is different. Right? There seems to be a consistency, and many children have visual memory deficits, but even the visual memory deficits are different within each child. I'll show you what that means in a minute. But as you can see, that each child is different. This is called the lack of a signature injury. A better example of this, these are 2 children that I evaluated. Both had very high IQs. The child, this column, is a boy. At this time, he was 16 years old. He had just failed out of high school. His IQ was 135. His sister was 15. Her IQ was 133. She struggled through high school, just made it out. She couldn't get to college. Right. Both children had problems with expressive language. The check marks indicate where there are deficits. The receptive language of both was normal. The boy had problems with his dexterity and motor construction. The young girl was intact. The boy had problems with impulse control. His sister did not. Both had problems with verbal memory. She also had problems with visual memory and visual working memory. With executive functioning, she had problems with cognitive flexibility. His was normal. He had problems with planning. Hers was normal. So you can see that even with siblings, their blood level levels, their peak levels were at the same age were about 27 or 28. The levels were very similar. The times of exposure very similar. They still don't look the same. This shows the lack of behavioral signature within an individual function, visual memory. The top graph shows recall performance, the short-term recall performance of a normal child. This is the figure. This is what the child drew. If you translate this into a score, it comes out at about the 75th percentile. The long-term recall also very high, and it's something called recognition, which means the child doesn't have to draw anything. They're just shown pictures. They have to identify things they've seen before. It all comes out normal. The second child, lead-poisoned child, you can see the recall is clearly abnormal. And scoring shows a very, very low immediate recall, low delayed recall, and very, very low recognition. This is a pattern that indicates that the information just never got in, or it got in and was immediately lost. The third child, also lead poisoned, you can see the recall again, very abnormal, but the recognition is normal. The free recall is not normal. This is called a retrieval failure, which means the information gets in, but you can't find it. You can't get it out. It's like if you take have a filing cabinet and it's alphabetically organized, you put the folder in the wrong place. It's in there, but you can't get it. You have no access to it. So even within an individual function, there's a lack of consistency from child to child. The lag effect. This is data from a child, a 7-year-old boy. When I first saw him, his verbal IQ was 72, third percentile. His nonverbal IQ was 101, 53rd percentile. His full scale was 84, which is kind of misleading. The full scale is really a meaningless figure. When there's such a discrepancy between the verbal and the nonverbal, the really full scale IQ is kind of meaningless. At any rate, when that child was tested at the age of 15, the IQ, verbal IQ, which was formerly 72, dropped to 56. The nonverbal dropped from 101 to 79. Full scale dropped from 84 to 62. If you look at the individual neuropsychological functions, again, the check marks are impairments. Expressive language was impaired and remained impaired. Attention the same. Impulse control the same. Visual memory got better, but then verbal memory got worse. Concept formation stayed impaired, and you also developed a problem with abstract reasoning. So this shows the lag effect, where things get worse over time. Effects on social emotional development. Remember, as pointed out by Dr. Schneider, there are problems particularly with the frontal lobes, which has to do with social judgment, impulse control, motivation. Basically, the social problems come from, and problematic behaviors come both directly and indirectly. Directly would be from the effects of lead on the frontal lobes and other parts of the brain, causing problems with judgment and impulse control. And indirectly, because children have academic problems leading to low self-esteem and other difficulties. So two things interact, and you wind up with really a toxic brew. And this has been shown in a relatively graphic way. This is from Rick Nevin. This shows lead exposure measured from, I think there's gasoline in the air with an 18-year lag between when you measure the lead and when you measure the murder rate. But you can see there's very, very close correspondence between lead exposure in the population and the murder rate. And here's one for lead and violent crime. This has been replicated over and over again. Schools fail lead-poisoned children because they're unaware the children have been poisoned. In cases where they were aware, they're unaware that it causes brain damage. There are problems when child's IQ is so-called normal. But the major problem is there's really very little access to neuropsychological testing. School psychologists are not qualified to do it. Most school systems have no access to neuropsychologists. So when you have lead-poisoned child and you know that they're brain damaged, if you don't do neuropsychological testing, any intervention you do is simply a shot in the dark. The odds are you're going to miss it, right? You're wasting your time, and you're wasting that child. So going back to this, primary prevention, we're trying. We haven't gotten anywhere near the goal. As far as the research goes, NIH is not investing any money in it. There's no political will. And as far as intervention goes, we don't have again, it's probably a matter of money. Ralph could speak to this as well. The financial resources to have neuropsychologists available to the school systems. So we're basically failing children on every level. Right? We really, this is a Jewish term called shanda. It means an ultimate disgrace. This is a disgrace. Thank you.
Unless there's some really specific trauma, almost every child referred has every symptom that could be lead poisoning. So how do we, on the behavioral health end, get them tested or even start discussing that it may not be just a child's behavior problem, there may be lead poisoning, especially years after their exposure.
The problem, that is the problem, because you need neuropsychology. Because Ralph actually speaks to, the school system. This is something that he's dealt with and tried to do, and I guess with a lot of obstacles. But that's the problem. That's why we're failing. It's expensive, and I don't think there's a will to do it. I mean, this is not a new problem. This is, we've known this. How many years? How many decades? It's a throwaway children. You look at the numbers. You lose sight of the fact that each one of them is an individual child, just like the one that Ralph spoke about, that 9-year-old child that was being taken away from some other. Each one of these is a human tragedy, and we don't seem to get there.
We also will be addressing this afternoon with the lead-related policies and things about how schools and health workers should be kind of better communicating with that later.
Also, I just wanted to add, the data from Rhode Island that I was showing that mean lead levels, and this is for the whole state, right, were, still 2 or 3. So that's the mean. So I think one takeaway from this conference is this isn't something that's only affecting a few kids. This is something that when you look at a kid who's behaving this way, you should think, well, maybe they have some lead poisoning, or maybe they have brain damage. And I think it does change the way that you think about the child when you realize that is a very real possibility affecting many, many children.
Yeah, but we clearly, and as you said, we're going to talk about this afternoon, but we clearly have to have a better way of identifying these kids from the outset.
Right. And having a way to communicate.
The speakers need to use a microphone.
Okay. Thank you. Okay. Well, we'll be addressing it this afternoon is what we were talking about. It's important to have identification.
Dr. Schneider, in your I have 1,000 questions, but I'll keep it to 1 or 17. In your experiment with the 2-back, where the kids were using more resources, did you happen to ask the kids at the end of that experiment how they felt? Did the kids who needed to have more resources, did they become more tired? Did they feel less successful? I mean, so if you have to use more resources, you can see a child giving out or just saying, I quit, it's just too much. I was just wondering if you asked the kid that.
We didn't, but interestingly, there was not a very good, and again, these were preliminary, so it wasn't a large number of children, but there really wasn't a consistent relationship between how much brain resource they were pulling in and their actual performance. So they could have been pulling in lots of resources and still doing poorly. I think this is more reflecting the amount of brain circuitry that was attempting to be recruited to try to do this. And they all tried. I mean, it's not that they gave up. I mean, they were all engaged and giving it a good try.
On the gender differences, do you think that there's some relationship between hormonal differences or an XY thing? Do you have any explanation as to why are there certain genes on the X chromosome that are being affected by the lead more than on the Y chromosome? your take on that?
Yeah, so I think, so we don't fully understand that at this point, but.
Can you repeat the question so that people can hear?
I'm sorry. Yeah, so the question was if we had an idea about what the basis might be for the sex-related differences, if it's hormonal or if there are certain genes that are on X chromosome or Y chromosome that are preferentially affected. And the answer is we just don't know yet. But from what we're seeing, it seems to be a combination of both those factors. That there are hormonal influences that are involved in lead preferentially influences some of those, and also there are some very specific sex-related genes that are also being affected. But we're just now starting to get to that level of analysis.
Yes, I'd like to know if the effects that you're talking about, does it matter how the exposure takes place, whether it's lead in dust or lead in water, in drinking water? And then, does this continue? We've talked about children very, very young, and the exposure coming from the housing stock, but does this continue as a child goes to school? Because a lot of school buildings were also built before 1978. Is the exposure continuing in the schools?
Well, in the work that we've done, we expose, it's all by ingestion of lead. So the animals have certain amounts of lead incorporated into their food so that they're ingesting it that way. The results that we get with that are not all that different from colleagues who are using lead in water as the source of exposure. So I'm not sure that it really matters whether it's from paint or from water or from dust. I think it's being ingested, and that's the critical point. And to your other point about do we need to be concerned about the lead exposures as the children are getting older and in schools and in older buildings. So I think that's another issue that hopefully will be discussed this afternoon, that I think not only do we need to be cognizant of the lead histories of these kids when they were much younger, but I think we really have to be paying attention to the concordant exposures, that exposures that are occurring at the age that they're in the schools, because that can be having an important influence as well.
Just to add a little bit more to that. As far as age goes, children under the age of 6 absorb more lead, retain more lead, put more from their brains. Right? That starts normalizing as the child gets older, but it doesn't really reach normal adult levels until the child's about probably somewhere in their teens. Right? So it's particularly dangerous for a child under 6. But secondly, the window of vulnerability opens up again when you're at 50 or 60 years old. And you start again, brain becomes start becoming more vulnerable to the effects of lead as well. So, the window's open to both ends.
I just want to add one thing, which is, the data we have is for testing children typically between ages 1 and 2. We really don't know what's happening to the children over time if they're just ingesting school water. We don't have the data. And many children who've been tested between at ages 1 and 2 move many more times in the course of their lives. We know in Trenton that 40% of children move during the school year. So if they moved into a house at age 3 or 4, we really don't know what's happening with their lead levels typically.
And the Coalition to Prevent Lead Poisoning in Rochester worked with the state school district. We did research, and San Francisco was the only school district, that had some type of lead law for their district, and it was very weak. So when you think about it, what happens in the school districts? When you had week vacations or long weekends, the custodians will come into a room, and if they're not using lead-safe work practices, what they're doing is they're scraping and painting. The children's books are there. Usually, primary children sit on the rug for small group classes. And we found that we were horrified with this because our custodians weren't aware of lead-safe work practices. We worked with the district, and now they're testing water fountains. They're making sure that lead-safe work practices are taking place. And this is extremely important because it's not dust, it's microparticles. And when the children are touching this, usually, many times, they don't wash their hands before they go to lunch. There may not be time or whatever. And so they're ingesting these microparticles. So it's extremely important to have lead-safe work practice and make that policy rock hard in school districts. And if you're in a school district, I think that is extremely important for you.
Thank you. Good morning, rather. I have a question about, can you discuss how diet can mitigate or prevent the impact of lead poisoning? I've heard talk about how fresh fruit and vegetables can mitigate some of the effects of lead, particularly with young children.
There seems to be increased lead absorption in a high-fat diet. It's important to have good calcium and iron intake. As far as fresh foods go, I'm not sure if they address that. But as far as things I do know, fairly well established, is that lead absorption is greater. For example, with fast food, high-fat food, and low iron or low calcium diet. Really important.
Hi, I just like to talk a little bit about nutrition. So the high-fat diets will enhance the absorption. Being hungry will enhance the absorption. Having low calcium, having low iron will enhance absorption in children. And many poor children both have high blood levels and iron-deficient anemia. So getting them into the WIC program is a really, really important thing. In addition to that, though, it's important to point out, there is no diet pill. You cannot give children a diet pill that's going to, provide all this fabulous nutrition for them and then not have to worry about them being exposed to lead in paint or lead in water or lead in some other source. So there are good reasons to feed our children well. Lead is one of them.
All the way back over here. How long should we be testing children for lead? How long should we be testing children for lead?
I mean, is there psychologically testing a child?
No, I mean, blood levels.
Blood level. That's sort of out of my area.
So can I answer some of these questions about lead programs? I used to be the director of the CDC's lead poison prevention program, and I have been in this business for 35 years. The reason we don't test school-age children is because we do have some data. It's population-based, national data. It can mask lots of small differences between small groups of children. But the average blood level of school-age children in the United States is 0.5 micrograms per deciliter. Doing wide-scale universal blood lead testing of those children is just not something that's going to happen. We need to test children who the state health departments are on the hook to develop criteria for which children need to be tested. They've done a really good job in New York, they've done a really good job in New Jersey, Massachusetts, Rhode Island, and plenty of other states. And they issue these reports both on their websites and to the local pediatric health care providers. So in Illinois, you would get a list of all of the high-risk zip codes in Illinois, and the law says all the children enrolled in Medicaid in those zip codes must be tested at 1, at 2, and at 3 if they've never been tested before, or if there was a problem with the previous test. That law is not being enforced, right? The states may also have a law that says we think that all kids, not just the Medicaid kids, all kids should be tested like that. Those laws are also not enforced. So that is really important. And then the second time that's important to do a blood lead test is when the parent or someone, the guardian comes into the doctor's office and says, my kid needs to be tested for lead because I work in the lead industry, because we just renovated our 1917 17, 20 farmhouse because for whatever reason, and I have never been to Princeton before, it's awfully nice here. It reminds me of Concord, Massachusetts, and all of those houses that I drove by, right? So we know that low-income children are most at risk, but the people that are living in those houses, if they have small children, are not immune.
Can I do a follow-up?
It's up to me.
So New Jersey does have universal screening mandate, and all children are required to get tested at age 1 and 2. Only 29% of children in New Jersey are being tested at age 1 and 2. And there has been a bill that has been passed universally that just passed last week, which is Senate 276. Last year, Governor Christie vetoed it. Hopefully he won't veto it this year, but it's imperative that it get passed. It's not just about lead poisoning, it's about point-of-care testing. But the reason that children don't get tested for lead poisoning in the state is because it is very hard to get tested, because the traditional way of getting a test is for them to, for doctors to write a prescription, and children who are most at risk for lead poisoning don't ever make it to the lab. So it is critical that this bill get passed. It's Senate 276, the Assembly side is 2332, and I encourage you all to encourage Governor Christie to pass this bill.
Can I just do a quick follow-up to that? Because I'm a public health nurse and I have been in Mercer County for more than 20 years. And the advent of HMOs in the state has decreased the number of Medicaid children tested for lead because they made it harder. Because the doctors used to be able to draw their own blood and then travel to the doctor's office. And there's been a dramatic decrease because they have to go to the lab and that's enforced by the insurance company. WIC used to test for things you're not allowed to test for anymore because of the HMOs.
Okay, let's go for the question right there.
Yeah, I feel obligated to make a couple of comments. I've treated thousands of lead poisoning children in New Jersey over the last 30 years. I disagree with the comment about testing. Testing never cures the disease. We have pretty good maps in the state. We know where the lead poisoning exposure occurs. We need action. We need to do something to prevent those children from being poisoned, not have to just test. The other thing is I can tell you that I also ran the medical component of the Institute for Child Study at Kane University. 50% of the children that were referred to us from learning disabilities have lead levels over 20. If we do believe that therapy, mitigation of their exposures and their lead levels produces some sort of plasticity, they get better. And I must tell you, as a physician, if I didn't believe that, I would have hung my hat up many years before I did retire in July. But we need to do something about that. So if you're asking, should you continue to test, I do think you should. If you have a child that is in your special ed, and you have a child that's having trouble in school, yeah, that child should be tested. The question that I'd like to ask you is, there have been some studies that I feel are somewhat unethical, looking at the effects of continued exposure of children in their homes, or even the use of a chelator to try to change that. Can you design an ethically sound study to solve the questions that you're asking? How do you determine whether interruption of the exposure to lead actually makes any difference? Maybe that's an unnecessarily answerable question. But there's been silence.
Can I address that, please? We've been studying the exposure of kids to lead for more than 25 years now. And we found over the course of the time, not just our group, but other folks, that cleaning interventions, remediation in the house, does in SIV studies, in pre and post, et cetera, makes a difference. Testing, I'll disagree. I think testing in kids tells us who's at risk and where the interventions may be applied. And even if it's not potentially for the child who is already high, their younger sibs or their future sibs, it may make a difference. So I disagree. I think testing in kids identifies who's at risk and who's not. And you can't do it by zip code alone, because some houses are clean and some are not, and you don't know until you get there and you test dust, and you test the kid, and you test the food, and you test all the potential risk factors, and you find out who's at risk and who's not. And then some of the interventions do make a difference in terms of post-identification exposure.
Okay, I'm going to tell you that we have to end at 11:45. We're very strict on schedule. I would unless our panel wants to say any last comments. Expusio wanted to say one last thing, and then we can let our panel, and then we will open up for more discussion during lunch and whatever. But Dr. Expusio, do you want to say something?
Very quickly, the targeted approach is what we did in Rochester, and we know the area, so it was a targeted approach and it worked real well. As a school principal, I asked the pediatrician, right when this was happening, I said, I want all my children tested. He said, well, wait a second here. He said, focus on the very young children, 3, 4, and 5-year-old children. That's where I got the data. He says, because here's what happens. The child can be exposed to lead as a toddler. And what lead does, it migrates from the blood and to the soft tissue in the brain, and then goes to the bone matrix and stays there. And so what happens is, he said this, when the brain is exploding in toddlers, exploding growth, that's the time that the child has been exposed. That's when major, at age 2 or 3, where major cognitive damage is done. He said, well, if you have a 5th grader who's showing symptoms of lead, that child the damage is done, has been done when the child was 2 or 3 years old. And if that 5th grader is not being currently exposed to lead, you're not going to find it in his blood. It's already in his brain and bone matrix. And the other thing that's really insidious is what Dr. Litsky pointed out, the lag effect. So that a child may not have a symptom at 4 or 5 years old, but yet when the child goes into 4th grade and has higher learning skills that they have to do, they hit the brick wall. So that developmental sequence is very complicated. And really, a neuropsychologist is really one that points that out.
Thank you very much. Is there any last statements for the panel? No? I would like to thank our panel very much.